We shall now talk about bacterial structure. What do the bacteria look like? This is a bacteria. This is a prototype bacteria. There's the outside of the bacteria and the inside of the bacteria. Inside of the bacteria is filled with like water mush, right? Water mush. That's called the cytoplasm, the jelly inside it. That's the cytoplasm. Then outside it, it has a membrane, the plasma membrane. Just outside the cytoplasm is the plasma membrane. But that's not sufficient because plasma membrane is not good enough for bacteria. Okay? It has to therefore have a cell wall. Cell wall is what prevents the bacteria from bursting. One of the fascinating features of bacteria is that their DNA is circular. Our DNA is like a string, long string. Our DNA is like a long string. Bacterial DNA is circular. You take string, like a circle. Take a wild guess. And then they have their chromosome. The interesting about bacterial, I forgot to mention one thing. You know the photosynthetic bacteria we just talked about before? Yeah? The cyanobacteria, the photosynthetic bacteria, they are thought to be the chloroplast of the future. The chloroplast. So bacteria, some bacteria came in and ate the cyanobacteria, and over time cyanobacteria evolved internally as the chloroplast. Wild guess. Chloroplast. Chloroplast. DNA. It has its own DNA. That's weird in itself, right? Chloroplast, it has its own DNA that's distinct from the cell's DNA. Take a wild guess. Circular, non-circular? Circular. Remarkable coincidence, huh? Okay. Now, so bacteria have circular DNA. It has one major circular DNA, and their tiny circular DNA could be floating around here and there, okay? But the main one single circular DNA contains most of the genes. And it has ribosomes. Ribosomes. The tiny little dot things are ribosomes. Ribosomes, the most important thing they do is they make proteins. They make proteins. How do you take amino acids and put them together in a long chain to make proteins? If you wanted to know that, you go ask who? Ribosomes. A bacteria has cell membrane, yes or no? Of course, they have. Every living cell has a membrane. Yeah, every living cell has a membrane. It's like the gatekeeper. If you want to get into if you ever wanted to get inside a bacteria, you have to go to the cell wall. Good luck. If you pass the cell wall, you have to go to the cell membrane. Double security. Double security. Analogy of a cell wall is the cell membrane. So this would be like cell membrane. Cell wall would be if there was a concrete like boundary around the building. You understand? If they did not have a cell wall, they would burst. Just leave it at that. In addition to that, so they have a cell membrane defense. A cell wall, for some bacteria have something called a capsule. A capsule. So if a French fry was a bacteria that has a cell membrane and a cell wall, right? If you ditch the French fry in ketchup, the ketchup would be the capsule. Do you get it? A third layer. These bacteria tend to be very pathogenic, dangerous bacteria. Those that have ketchup, those that have the ketchup, those that have capsule, right? All bacteria have cell membrane. All bacteria have cell wall. Yes, all have cell wall. They have cell membrane, they have cell wall. All bacteria have capsule. All bacteria have capsule. Not all of them do. Some of them do. Some of them do. From inside out, plasma membrane, cell wall, maybe a capsule. Pathogenic means causing pathology, illness. So illness to us. To them, they're just defending their territory. All right. There's a video on this. There are two types of bacteria by the way they look. By the way they look. Okay? So, these are called, why is this important to us? Okay, this is important to us. Okay? So they're called gram-positive and gram-negative bacteria. Okay, the way they look. Gram-positive bacteria, okay, you'd rather not have any bacterial infections. They can both be deadly. But gram-negative ones tend to be more commonly deadly. You follow? So somebody goes to the hospital with fever, they take a blood sample, take a blood sample, and then they put some of that blood sample on those petri dishes. You know what petri dishes are? Petri dishes are what? You know what I'm talking about. Are they empty? No, they're not empty. They have lunch there for the bacteria. Otherwise, how else are they going to grow? How is the bacteria going to divide if they don't have any nutrition? It's like impossible, right? So a petri dish is a lunchbox for the bacteria. Yes? No. Yes, I'm just checking. You're just, because I know you're not paying attention. Focus. So a petri dish is just lunchbox. So you put it in there, they're like, oh my God, it's heaven, let's divide. You know? Yeah? So they divide. And then, so you take a blood sample, put it in there. It takes time for bacteria to divide. Patient has gram-negative bacteria growing in their blood, meaning that they're growing in the petri dish. They have gram-negative bacteria in blood. Serious stuff. Okay? The difference is this. Gram-positive bacteria, this cell membrane, you see this? That's the cell membrane. All bacteria have it. And then there's a cell wall, right? And then and then there's a capsule, some of them have. Now gram-negative one, cell membrane, cell wall, and cell membrane, and then maybe a capsule. So now the difference between a not so bad dude and a really mean dude is what? One extra membrane. One extra membrane. Now the amount of cell wall, the cement so to speak, that the bacteria has, gram-positive bacteria has, is more than the gram-negative bacteria. In other words, who really makes the bacteria tough? The membrane or the wall? The membrane. They're both important, don't get me wrong. But membrane is selective. Means you really have to have the right keys to get inside the lock and get inside. Do you understand? They're harder to kill, therefore. Many bacteria that we have, okay, are killed by antibiotics that have to go inside the cell. Inside the cell. This gram-positive, gram-negative, what's gram mean? It's not gram like you weigh gram, like 50 gram. No, it's the guy's name. It's the dude's name who invented the stain. Okay? So there are two types of bacteria based on how they look under the microscope. There are different ways. There are different ways the medicine gets inside the bacteria. Okay? And they mess up with the bacteria stuff into like ribosomes, like azithromycin. Like azithromycin blocks the ribosomes. How come the azithromycin, okay, it kind of messes up all the bacterial machinery by blocking the ribosomes, okay? So how come we don't get sick? How come it doesn't mess with our ribosomes? We have ribosomes too, you know. What do you think? What'd you say? Selective. It's exactly true. Bacterial ribosomes are different than human ribosomes. Hopefully, oh my God, we have so many, yeah? They're different ribosomes. So they only affect the ribosomes of the bacteria. Until then, Assalamu alaikum.